So the next broad topic that we're going to want to talk about over the next couple of weeks is the question of genetic engineering. Now again, before we dive into the specifics of this topic, I want to show you an external video. And this is a video, it's a little bit longer, it's about 20 minutes long, and I realize that makes our whole presentation a little long this week, but we can shorten it up a little bit next week. But it's really worth watching, and it's from the Human Genome Project, and it describes the Human Genome Project, which was an effort to kind of decode the whole structure, the whole sequence of the human genome. There's some really useful information in this video about the nature of genetics, the nature of genetic sequencing, of the human genetic inheritance, and some things at the end about kind of the possibility of genetic medicine, which includes some of these possibilities about genetic engineering. So I'm going to pause here and let you watch that, and then I'll come back and talk about some of the issues that this raises.
The task we're up to here is figuring out how life works.
At a very, very fundamental level, it's a key to understanding our species.
Everyone has a genome. We are all human, and it's the basis.
It is the word that means all of the DNA, all of the hereditary material of an organism.
The DNA is the script for life.
It's the stuff that tells one gene to make its machine in muscle cells and another gene to make its machine in brain cells.
In fact, that DNA code represents solutions evolved by life 3 billion years ago in some cases.
The human genome is essentially our shared inheritance. Bacteria have their genomes, and dogs have their genomes, and plants have their genomes, and we have our genomes.
Our DNA is almost identical with chimpanzee DNA. It's only about 2% different. Now, people probably aren't surprised that we share our genes with monkeys, but it turns out that we share about 50% of our genes with a tiny little roundworm. Presumably, a billion years ago or 500 million years ago, we had a common ancestor with the worms. And what we learn about those genes in worms and flies and yeast, now we can translate back to humans.
So we are just another species on this planet.
We are a very small species. The entire human race traces to a population of an effective size of only about 10,000 people in Africa, perhaps 150,000 years ago.
And because we're a very new species, we ain't been around that long, we actually share most of our DNA at a remarkably identical level.
We are all 99.9% identical. Of course, 0.1% difference out of a genome of 3 billion letters means any two of us also differ in 3 million separate places in our DNA.
Yeah, the bacteria won't come off right away. You might have to twirl a little. You might have to...
We all think of ourselves as completely unique individuals. And at some level, we are.
If you've seen identical twins, you've noticed that they look a lot alike. Well, they have exactly the same DNA between them. When you get to know them, you'll also find out that they're different in pretty significant ways.
But it's important for us to recognize how connected we are, how close we are with every other mammal, having the same sets of genes, the same body plan, how close we are with trees and funguses, which run their cells pretty much the same way we run our cells. powerful way to see this grand unity of life is to read the DNA code. It's a 3 billion letter text.
If we printed out DNA sequence and put it in a book, you'd turn the pages, and what you would see would be a funny language, where there are only 4 letters in the alphabet, A, C, G, and T. Those are the 4 bases, as we call it, of DNA. It would take many of these books stacked on top of each other, about the height of the Washington Monument, in fact, in order to represent all of those 3 billion letters that you have inside each cell of your body.
And yet in that, those strings, in exactly the order of the letters, lies all of the fundamental secrets, lies all the instructions that our bodies use to build themselves from a single fertilized egg, the whole thing, the whole Taj Mahal of our bodies.
What we have when we want to analyse a genome is a huge problem.
Well, in the mid-1980s, a few people began to imagine that we might mount a carefully orchestrated attempt to read the entire sequence of the human genome.
The thought of sequencing the whole human genome was utterly outrageous when it was first proposed in 1985. People could barely sequence a couple of hundred letters of DNA at a time. It took a tremendous change in technology to make that possible.
You discover, that there are pessimists in this world, and, you spend your time with optimists.
What genomics is about is taking a global picture of all of the cell, all the DNA instructions, all the RNA readouts of the genes, all the proteins at once.
It's like, for example, mapping the continental United States. At the DNA sequence level, we certainly want building by building this whole map. We want yard by yard across the whole continent. We take the big picture, we divide it up, and we organise the smaller pictures relative to one another.
And again, congratulations to our colleagues in Germany and Japan.
The Human Genome Project is an international, multi-component effort to read out the instruction book for human biology, that is our genome, all of the DNA, and to understand what it's telling us.
We had to build all sorts of automated robotic systems to process hundreds of thousands of samples a day.
We work with about 1,000 plates of DNA here a day, or 100,000 DNA molecules sequenced here a day.
DNA starts at one end of the process in the lab and works its way through all the way down these hallways behind me as a large fragment of DNA.
The process of sequencing is determining the sequence of all the individual units in the DNA.
As it moves down the hallway, it gets chopped up into small bits, turned into many smaller bits of DNA that work their way through all of these robotic lines to get purified, sequenced.
It is a process that has to be repeated many, many, many times because of the size of the human genome.
And eventually hit a sequencer, which will give you the fluorescent readout of every base pair in that fragment of DNA.
Whilst we can get a sequence of bases in the genome, we don't know everything yet about how the bases are organized.
We're just beginning readers right now.
We have only the faintest glimmer of what a lot of this information is really telling us.
In a sense, we have the book of human life. We can read the letters, and now we're trying to find the words and put them together into sentences.
We are making this information completely available, automatically available nightly. It's just picked up and automatically sent to public databases.
This means that any scientist anywhere who has some idea can use this information to advance the question that they're working on. And this suddenly empowers all the brains of the planet.
So by putting it out daily, we're speeding discovery.
It's something about a big project where you've got a lot of people working, real synergism, where the sum of the parts is more than the individual components.
It's giving biologists an opportunity to truly understand what's going on inside living systems and look at the parts that are actually moving inside and to understand how they are failing to work properly when they fail.
We wanted to have it to cure disease.
It's cloudy, but you do have some fluid up in here, so we were thinking
We will now be in a position fairly soon to understand what underlies that otherwise mysterious and threatening reality, that some of us are strongly defended against certain threats and others are vulnerable.
Genes in general are there for very good reasons. We'd be in bad shape without them all working together to keep us healthy.
Just relax and breathe normally.
But if there's a glitch there, in a particular one, then it may not quite do its job, and then a disease may result. A certain set of illnesses, things like cystic fibrosis or sickle cell anemia or Huntington's disease, a particular kind of misspelling, almost certainly you're going to develop the disease. But many diseases aren't like that. The variation, the misspelling in the gene increases your risk, but it's not yes or no. A misspelling is simply a way of saying, oh, you got a G there instead of a T. Or you had a GTCC there and now it's just GCC.
We will be able to predict those things to a very high degree once we are able to go in and say and ask the question, which versions did you inherit, Albert, as against some other person?
Now we all have these. There are no perfect specimens. We're all flawed at the DNA level. We have probably 30, 40, 50 glitches and that are in places that we wish they weren't. So a lot of the focus is going to be on prevention. How do you keep people from getting sick in the first place?
Then in time, therapies, medicines, etc. will come along and it will be of great benefit to everyone.
I think it's very hard for most people to really grasp how fundamental and how revolutionary knowledge that's coming from genetic research is in terms of the decisions that they will be making in the future.
We're not talking about something that's distant from people. We're talking about things that are going to affect your reproduction, your health, your daily life.
You're going to get knowledge, and how do you deal with it?
When I get the chance, when I talk to people, I tell them that this information is coming and people have to know about it.
I think any kind of new knowledge is frightening until we understand it more.
We know that there have been bad experiences with people misinterpreting genetic information and using it for ill purposes in the past.
So we're engaged in a very interesting experiment. Is it in fact possible to study the ethical, legal, and social implications of a scientific revolution at the same time the scientific revolution is happening? We haven't, as a human race, had a particularly good track record there.
I think any project that has the capacity to affect so many lives, for good or for ill, has a fundamental obligation to study what is good and what is potentially wrong.
We can't be so afraid of it that we don't allow ourselves to use it to correct things that really cause undue human suffering.
What does it mean to understand the sequence of the human genome? Are we in danger of beginning to think of ourselves as marionettes that are manipulated by strands of DNA? And is this going to damage our view of ourselves as creatures of free will and spirit?
I think it should be obvious, and it isn't obvious, that we are far more than just our genes.
We're clearly a complex mixture of what our genes are telling us and what the environment does to us.
But I think as human beings, there are aspects of who we are that this sort of approach based on this chemical called DNA is probably not going to reveal the mysteries of. We won't understand a lot about human relationships. We won't understand what love means. We won't understand the aspects of us that are more spiritual than mechanical.
We've been trying to do science without even knowing the parts list, without even knowing the building blocks of a human.
But this is, I think, one of the most exciting moments in all of science, this adventure that we're on.
I'm sure 100 years from now, people will have better lives than they now do.
When I have an opportunity to sit back and think about the impact that this project is going to have on science in the future, I feel very excited and very proud to be part of it.
The rate at which we advance in trying to ameliorate human suffering will now go up extremely steeply.
And the kinds of moments that are comparable in my mind are big moments in history, when Copernicus and Galileo discovered that the Earth was not the center of the universe. About 150 years ago, Darwin showed us our place in the animal kingdom. I think what's happening today is that we're finding out now at an internal level much more about what we really are.
This is humanity, this is Homo sapiens. This is it, this is the secret of our lives.
So a number of the issues that genetic engineering, genetic science raises are mentioned in this video. One is the question of pre-birth screening. So the possibility, without destroying the embryo, destroying the baby, to see, to do a test to see if there are certain markers present that might indicate certain kinds of problems or diseases. Now, ideally, we would say that that would give the opportunity for the doctors to correct some of those things. But of course, what often happens is that the option is given, there's really no way to correct them, but the option is given to terminate the pregnancy, to have an abortion. And that, of course, raises ethical issues that we'll want to talk through. There's also the possibility of, I really should have up here kind of preconception screening. So there's certainly the possibility for a man and or a woman to go through different kinds of genetic counseling that might suggest the possibility, if they do conceive and give birth, of different kinds of issues arising. And that, again, will raise a set of ethical issues that we'll want to talk through. So another category that I have up here is genetic enhancements. And this is the possibility of a fetus developing in the womb and its genome being modified in a certain way, or after a baby is born or when a person is older, in some way the possibility of its genome being enhanced or modified, not to correct what's considered a disease or an abnormality, but to change something in a way that's perceived to be better than what would normally be the case. So, maybe you want a baby with blue eyes. Maybe you want a baby that's likely to be 6 feet tall. Maybe you want a child that's likely to have really great physical strength. Maybe you want a child that's going to have an above average IQ. Now, right now, most of those things we really can't do, but the technology certainly could be there that we could do many of those things. And the question then arises, well, is this something that should be permissible? Is this something that should be acceptable? What's the line between saying, well, I know that there's a certain genetic marker for a kind of deadly disease, and I can repair that in utero. What's the difference between that and saying, if this person's 6 inches taller, they're likely to have a much more satisfying and productive life? Why is the one considered a correction and the other considered an enhancement? Why is the one the correction of the disease probably not as ethically concerning to most of us as the enhancement? Those are issues to think through. Post-birth genetic therapies. So this wouldn't be, enhancements in terms of changing part of the genetic code, but using therapies that treat certain conditions. There are now cancer drugs that are really genetic therapies. There are screening methods of trying to determine whether someone is likely to develop a certain kind of cancer. And again, you might think, well, that doesn't sound that ethically concerning. A lot of the ethical issues around those things tend to be who has access, tend to be, is the screening really likely to show something that's likely to happen or just to raise unnecessary fears? We'll talk next class when we talk about some of the legal issues about the BRCA genes, which is the genes that show a susceptibility to breast cancer. And there was some legal cases around the patents on the BRCA genes. But one of the questions there is, it doesn't mean you're going to get breast cancer. It just means you're more susceptible. So do we really want to tell people this? Because will they go out and get an unnecessary mastectomy or something along those lines? We can talk about those. And then another cluster of issues is this question of posthumanism. What does posthumanism mean? Posthumanism is the idea that it will be possible to genetically enhance and genetically modify human beings to the point where they effectively become a different species, something beyond the present notion of humanity, maybe even technically a different species that is really not capable even of sort of crossbreeding with the current human species. And if that is possible, is that something that should be considered potentially ethical? Is that something that should be considered really dangerous and unethical from a theological perspective? What might that mean? I mean, if Jesus became incarnate as a human being and died for us, what does it mean if we become post-human? Is that even possible? Raises a number of really interesting questions that we can and will talk about. Okay, our final cluster of issues is the question of assisted suicide and eugenics. So the idea of eugenics is the idea of selecting among the members of a species or a group, here of course the human species, to select out certain kinds of people and select in other kinds of people. Of course, the classic horrible historical example of this is Nazi Germany, which had an active eugenics research program in an effort to try and kind of purify the Aryan race. And of course, in the attempt to wipe out the Jewish people in the Holocaust is also kind of part of that really twisted idea. So without doubt, there are huge ethical concerns that almost everyone agrees are issues when it comes to the broad question of eugenics. But when we drill down into some more specific questions, and in particular when we tie it to some of these genetic research issues that we just looked at, things can become a little bit more difficult. Let's say that it became possible to repair the genetic conditions that lead to Down syndrome in utero. And effectively, in doing that, to be able to eliminate Down syndrome. Now, there's a sense in which that's a form of eugenics. There are people who, today are living with Down syndrome. And is it appropriate, is it ethically appropriate to say it would be good to eliminate that, to effectively eliminate that group of people? Is it saying, that's a condition, sort of like it's an abnormality, kind of like a disease that it would be good to eliminate, or is that something else? Those are real difficult issues, and we'll talk about some of those. When we come to assisted suicide, of course, we have the general issue of should any individual person be able to decide that they want to end their life at any point in time, whether it's because they're suffering from a very painful and difficult medical condition, because they're suffering from a terminal illness, or simply because they no longer desire to live. Is it ethical for a person to make that choice? Is it ethical for a medical professional or anyone else to assist them with that choice? Is this something the law should allow or prohibit? We'll talk about the legal issues next week, and as you know, those are difficult legal issues and political issues that are kind of in the news regularly. Some of the more subtle questions really relate to what I have up here on the screen as extraordinary measures. So, what if someone is on life supports? When is it appropriate to terminate those life supports? What if the life supports include not only artificial respiration, not only keeping the heart and the lungs going, but also artificial nutrition and hydration? Is it appropriate to turn off the heart and lung machine and also turn off the hydration and nutrition, or do you have to keep feeding the person? These are issues that have worked their way through the courts and that are ethical issues for us to think about. Another one is who may consent. Is it necessary for the person themselves to either consent or have given advance consent, or can a family member consent, to turn off the machines? Those are difficult issues. And then a final term that I want you to have that we'll think about is the principle of double effect. So the idea of the principle of double effect is that there are some treatments that are given, particularly or often pain management kinds of treatments, that can also have the effect ultimately of shortening or limiting the person's lifespan, or potentially even terminating the person's life. So if you think about someone, for example, who's in an enormous amount of pain, maybe suffering from a terminal cancer and is in a great deal of pain, you could give them a very large dose of morphine, and the morphine would manage the pain, but it also has a very good chance of ceasing their respiration and thereby causing their life to end. Is it acceptable to give them the morphine even though there's a very high risk that it will kill them? That's the principle of double effect, and very often that principle is used to talk about, when is it appropriate to do certain kinds of measures that may terminate a life, but that also have a different effect of mitigating pain or mitigating symptoms or other things along those lines. So that's another one of the main issues and a term that you want to have as we think through these things.